together at Minnesota at one point. I promised myself that I would not tell stories about them, although I could if you really insist. Anyway, today I want to talk about cell motility, and I'll start off by, I think I will, there, by indicating that cells can move in very, very different ways. On the left I have a bacterium, about a micron or so long, swims by rotating flagella that are embedded in its membrane. And if you, there's supposed to be something is not functioning quite correctly. There. If you look at it, what you see is they do straight line runs punctuated by tumbles, which occur when the bundle of flagella flies apart. So in the bundle of flagella, there are 6 to 8 of them on the cell. When it's wrapped together and rotated counterclockwise, then it moves in a straight run. Otherwise, it tumbles, essentially doing a random walk at a point in space, chooses a new direction, and then moves off. On the other hand, some cells crawl. And the canonical picture of a crawling cell is one that has some attachments to the substrate. It protrudes part of its leading edge, attaches that to the substrate, and then somehow pulls up the trailing edge, and by that process moves. And if you watch this leukocyte, which is chasing a bacterium, one of the things that I want you to notice in particular is how much this cell deforms itself as it's twisting and turning, trying to catch the bacterium. So what that means, of course, is that the cell is undergoing, this is a single cell, the cell is undergoing vast internal changes in its structure to be able to deform itself so as to move in this way. And of course, the denouement is that the bacteria gets eaten. Now, a question is then, do cells that normally crawl also have other ways of moving? This cell, you'll see, attaches very strongly to the ECM because you can see how it contracts the ECM. But on the other hand, some cells that normally might crawl can actually swim. The experiment here is that you take cells and you put them in a fluid with a variable density so that they become neutrally buoyant at some height above the bottom of the petri dish. And then you put in the micropipette, which is what you see here, and put in an attractant at that point. And then you see the cells, you can see collectively moving toward that signal, but they don't have anything that they can attach themselves to. So the question is, how do they move? And the answer is, they swim. And when you watch how they swim, you see the enormous contortions that an individual cell undergoes as it swims. Watch this guy, for example. Look at the shape changes that it's undergoing as it's swimming toward the attractant at the upper point. So these things, and dictyostelium, which is one of the canonical systems that's used in cell motility, differentiation, and a variety of other ways, actually has a variety of different ways in which it can move. It can do what is called gliding, where it essentially sticks out a leading edge, a pseudopod, etc. It can do what Peter van Hastert here calls walking, where it's a combination of sticking out the front and attaching it and pulling the rear back, and simultaneously propagating little protrusions down the side. And then the third motion is to forget about the pseudopod completely and just move by propagating the little protrusions. Okay, so this raises at least 3 major questions. The first one I'll call the exterior problem. How do the shape changes actually give rise to motion? Okay? And you saw in dictyostelium how dramatic those shape changes could be. You saw in the case of the neutrophil that it had to undergo significant changes in shape in order to track the bacterium. There's the interior problem. That is, how does the cell actually control the shape changes? I mean, these are not just random shape changes that it's undergoing in order to move. Somehow the cell is controlling them, and when there is an attractant in the environment, the connection between the exterior and the interior problem is the transduction problem, where the cell receives a signal on the outside via receptors, etc., somehow transduces that into an intracellular signal. By the way, if somebody wants me to slow down talking, by all means say so, because it might be difficult for people that are not quite so familiar with English to understand me. Am I going too fast for people? Don't feel bad if you say yes. Okay.
Only Koreans can say yes.
Pardon? What?
Only Koreans can say yes.
I don't know. You can say yes if you like. But anyway, so it's the transduction problem that connects the exterior problem of the shape changes and the motion with the interior problem. So how do you do this? So I'm going to look first at the exterior problem. And what we're going to look at is an object swimming in a viscous Newtonian fluid that's incompressible. If we write down the Navier-Stokes equation and scale them in an appropriate way, you get this equation, where this quantity is the Reynolds number. And if you look at the Reynolds number for some typical kinds of swimmers, you see an enormous range. A large whale swimming at 10 meters a second is something like 3 times 10 to the 8th, whereas at the other, which means, incidentally, that of course the inertial effects, which are represented by the terms on the left, it's the inertial effects that dominate a whale. They also dominate you if you swim. On the other length scale, though, a bacterium, which is of the order of micron and can swim at 10 microns per second, the Reynolds number is 10 to the minus 5. So what that means mathematically is that that left-hand side is essentially unimportant, except perhaps for very dramatic changes in velocity. Okay? So U, as I forgot to say, but I guess it's obvious. U is the velocity of the fluid. Okay? So the problem then is what's called Stokes flow. For a low Reynolds number flow, you have to solve this equation for the velocity and the pressure. And this is the incompressible condition. Divergence of the velocity equals 0. And the first thing you notice is that time does not appear in this problem. Okay? The time has disappeared, which means that the motion, if you have a motion u comma p, a pair, then minus u comma minus p is also a pair. Okay? And the implication of that is that not all motions will produce, or not all shape changes will produce motion. In particular, the question is, when can the swimmer propel itself by shape deformations? And it's much easier to say when it can't. And this is the result or the statement of a famous theorem called the scallop theorem due to Purcell in the mid-70s, which says that if the sequence of shape changes is cyclic and time periodic, if it's identical when you view it under time reversible, then there will be no motion. In other words, the origin of the scallop theorem, of course, is the scallop, where you think of it opening and closing its shell, all right? And by the theorem, it should not move. But fortunately, scallops have not taken any mathematics because they move rather well, in fact. If, I don't know what's wrong. There it is. There it comes. This is bizarre because this thing worked earlier this morning. Spaceships. Is it behind it? Oh, it ended up behind. Okay. So watch this guy. So starfish prey on scallops. They basically wrap their arms around the top of the shell and pry them open and then eat them. Okay? And so this guy moves very effectively, and he moves in exactly the opposite way you would predict if the motion is due to the opening and closing of the clamshell, because then you would think he would move backwards. But in fact, what they have is a series of jets along the back side here, where when they close, they push the water out of those jets along the back, and that's what moves them. So anyway, So here's the swimming at low Reynolds number problem. Okay? You have an object whose shape you're going to deform periodically in time. All right? That means, in effect, that you're specifying the velocity on the boundary of this object. Right? You want to solve it subject to a cyclic swimming stroke, which simply means it's going to undergo a periodic sequence of shape changes. Okay? So think of it in very simple terms. Pushes something out, pulls it back. Pushes something out, pulls it back. That's not going to make it swim, but that's the way to think about it in simplest terms. And the canonical problem then is to solve this subject to the fact that the object is force and torque free with a velocity specified on the boundary and the velocity at infinity equal to 0. Okay? So that's a choice. Okay? That's a choice because the moment you put it in a finite domain, the object has some interaction with the boundary. But specifying that the velocity be 0 at infinity makes the analysis a good deal easier. Okay? So then if you give the object an arbitrary sequence of periodic shape changes, that in general will not be an admissible motion because you will have forces and torques. In other words, it will produce a non-zero velocity at infinity, but that's exactly what then allows you to compute your translational and rotational velocities. You take a Stokes flow at infinity with exactly the counter velocity that's needed to produce 0, and that gives you the translational velocity of your object. Similarly for the rotation. That may sound a bit obscure, but I think it'll become clear when we go through this. Okay? So here's the algorithm. Solve the Stokes flow with specified shape changes. Look at the asymptotic behavior at infinity from the conservation of momentum, and then take off exactly the Stokes flow that corresponds to a rigid motion that gives you 0 at infinity. Okay? So an abstract way to formulate this, and this is a nice way to think about it, but it's not very useful from a computational standpoint, is the following. You think of the motion and the shape as living in a fiber bundle, and the base space of this bundle is the shape changes, and the fibers are the Euclidean motions, and the question then could be phrased geometrically. If I go through a periodic sequence of shape changes in shape space, can I do a net motion in my Euclidean motions? Okay? That's a nice way to think about it, but computationally it's not very helpful. Okay? So Purcell came up with a swimmer that could do it. The first result, or the first consequence of Purcell's theorem, is that you need at least 2 degrees of freedom in your object. All right? So the clamshell, with its 1 degree of freedom, if that's all it was doing, could not move. So he devised a little thing that has 3 rigid pieces, the end of which can flip back and forth, and showed a sequence that could lead to the shape changes, and then other people came along and computed them. And if you watch this, you can see going through a cyclic sequence of changes actually produces motion. Okay? Now there's some real approximations in doing this, but when I show you the real problem that we work on, those will disappear. But this demonstrates that Purcell was correct in his analysis. Okay? So what Xishuan Wang and Jiefeng Hu and I did a couple years ago was develop a 3-sphere model in which, to give you kind of a canonical idea of how shape changes can produce motion without doing the computational analysis that we'll need for a real problem, because this one can do a lot analytically on. And the idea is the following. You start with 3 spheres in a line, and these spheres exchange volume with each other. So they maintain a certain distance between them, but then they exchange volume. And the question is, can you devise a sequence of volume exchanges which produces net motion? And the answer is yes, else I wouldn't have shown this. what you do is start with them in this position with the center one large. You put a little bit of volume back into the left one, which moves the whole ensemble to the right a bit, and so on. You go through that sequence of shape changes, and here's a illustration of that. If you watch the projection at the bottom, you'll see that this whole ensemble is actually translating to the left. Okay? Again, there are approximations in this because I'm going to show you a little bit of analysis in a moment, but it demonstrates that an object, by rearranging its shape or by extension its mass, can actually swim. Okay? Pardon? Zero. These are all zero because it's Stokes flow. I mean, you might ask, how can it be truly zero if there's motion? And the answer is, of course, it can't be truly zero, but it's to within something which is of order the Reynolds number. Okay? That's why I said at the outset when I showed you the Navier-Stokes, the left-hand side is irrelevant except if you undergo rapid velocity changes. Okay? So if the object is doing rapid impulsive motion, then of course you have to look at higher order terms. Okay, so one does a little bit of analysis, and I guess I won't go through this in any great detail because Philip is a hard taskmaster. But the point is, you can approximate each of these volumes by what's called a Stokelet, a point, the flow generated by a point force, and then neglecting the interaction between them, so they have to be far enough apart to do that. Then you can actually write down the solution for the velocity for each of them, where this is the rate at which their volumes change. Write out the solution for each, apply the force-free condition, etc., etc. And you can do things like compute the power needed, the efficiency, etc. And these objects are what we're interested in because we want to know how efficient is a given sequence of shape changes for producing motion, for example. Anyway, so here are just a couple of implications of that analysis. So the first one is that if you only shift volume between one pair at a time, then you can always predict what the direction of motion will be. Okay? And I showed you what it was in the previous case. If you look at strokes, so now there are 2 degrees of freedom here, the exchange of volume between the center and the 2 peripheral ones, so that if you have a stroke homotopic to a circle in the plane, you increase the stroke amplitude and you'll increase the translation, etc., etc. So these are interesting things to predict, but they don't solve the big problem. The next step towards solving the big problem is to look at a 2D flow. And the reason for doing a 2D flow is that for Stokes' equation, we can use complex variable methods to make a lot of progress before we actually do computations. In 3D, you're almost immediately confronted with having to solve the problem computationally. But here we can make some progress analytically. And again, the idea is start with this set of equations, Stokes equations and the divergence free. Those are equivalent to the vorticity and the pressure being harmonic. That means you can introduce a stream function that satisfies this equation. That defined this way and satisfies the biharmonic equation. And the nice thing about that is that the solution of the biharmonic equation has been known since Goursat's time. Okay, so you know exactly what the structure of the solution is, and it can be written this way. The imaginary part of a function phi plus a function chi. All right, so you know what the structure is, and if we can find the functions phi and chi, then we have it. Okay? Of course, you're interested in the actual physical variables themselves, pressure, velocity, vorticity, etc., and you can get all of those from that. And this was done earlier in the context of elasticity back in the 30s and 40s by some Russians, although I gave this talk a while ago and there were some Ukrainians in the audience and they took some note of the fact that Muscovich, whatever, was not a Russian. Anyway, okay, so what do we do? Well, look, in the context of elasticity, the problem you solved was, given a domain, say, with a hole in it, how do you predict the stress around the hole? But in that situation, you had a fixed geometry. What we're trying to do is take an object which is changing its shape periodically, find the flow exterior to it, and then predict things like the velocity, etc. So how do you do that? Well, that's where the complex analysis really comes in. So you can always map a shape like this, which might be one step in the sequence of shape changes of the object, back to the unit disk, all right? Solve the exterior problem there, and then map forward. So by mapping back and forward, you can do that. And you do that at a sequence of time steps throughout the period of the motion. So if you're given functions over here, then you have to find functions. Well, first of all, you're given the velocity on the boundary of this object in the z-plane. And if you map that back, that means that that velocity looks something like this. It's complicated, but and obviously you can't absorb all of this at this point. But the main idea is that there are these two analytic functions that we're trying to find, which describe the flow. And we have to use some other techniques from complex analysis to do that, because we want a function which is defined both interior and exterior to the object. There's something called the Plemmel formula, which allow you to do that, and then you do the approximations to the interior, and the exterior velocities. And then you realize there's a problem, because we have this one boundary condition which involves two unknown complex functions. How do we get that? Well, the way you get that is apply this operator, which looks a bit like a Cauchy integral without the argument, and you apply that to this velocity boundary condition, and you end up with an integral equation in only one of the unknown variables. Okay? So in that way, you're able to convert the problem to something which is solvable. If you look at conformal maps with only a finite number of terms, then this integral becomes a sequence of linear equations which you can solve, and the problem is reduced to relatively simple numerics. Okay? So, what are the results? Well, let's look at a sequence of deformations, shape changes that look like this. All right? So, some of you may remember. How many remember what a Joukowsky transformation is? Okay, I didn't really expect you to. But a Joukowsky transformation is something like zeta plus 1 over zeta, which can give you a nice airfoil shape, for example. Okay? So the point is that complex transformations like this can generate a variety of shapes that are relevant to motion. And if you remember what all the things we're interested in, rotation, translation, mean velocity within a period, the force distribution, etc., 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 all of those can be extracted via solving that series of linear equations. Okay, and here are one of the results. So suppose let's go back. Suppose you fix the phase change in here, and you randomly vary the amplitudes of these different modes. All right. If you say take a swimmer with 3 modes, one thing that immediately comes out is that there are no motions out here with an efficiency that is significant. Okay, you see a very sharp cutoff in the relation between the variance of the power and the efficiency. If you go back, I didn't stress this. The variance of the power is some measure of how much your power is changing during the swimming stroke. All right? And you see that, for example, if you put in a force that varies very dramatically, you will get a big variance. And these results show that if you have a big variance, you have a very low efficiency. So the first conclusion is that efficient swimming should involve a relatively gradual sequence of shape changes. But then you think back to dictyostelium and the ones I showed you, and they were anything but nice and smooth. So one could probably conclude that they are not swimming very efficiently at all. But let's see what we can do that's a little bit more relevant to dictyostelium. Here we go back to complex analysis again, and we recall, or for the first time see the fact, that you can write explicitly conformal maps between a polygonal shaped domain and the unit disk. Okay? Before I was taking these shape changes that were generated by 1 over zeta powers. Here I'm just going to take a polygon, find the Schwarzkopf transformation that maps it back to the disk, solve the problem on the disk, and then do the reverse again. Okay? So you start by building these up. So what we did, or what Qixuan did, was you start with a linear segment here, a linear segment here. That's part of the polygon. Then you put on these sort of almost hemispherical, well, it's part of a column with a hemispherical cap on it, and you make polygonal segments around the cap. The point is that you can define a polygon which approximates some of the shapes that you see during the movement of dictyostelium. And then you want to ask, as I change those shapes dynamically, what's the effect on the swimming? Can they swim? Do they not swim? Etc., etc. Okay, so here we are. Remember, this is the time snapshots of the shape changes. And two different people looked at these, von Hastert and someone named Barry. And here are the kind of average scales that you see for later references. Okay, maximum body lengths of the order 20-25 microns, body widths 5 microns, etc., etc. And one question is one or the other of these swimming more effectively? Can both of them swim? And this little video has one of the answers. So here is one of those swimmers. Here's with n equal 15 points in the Schwarzkopf, n equal 15 vertices is the way to say it, or 50. And you see the smooth swimmer moves very effectively. Okay, so you could think of this as a way to devise a little robot that can swim in a blood vessel, for example, by these shape deformations which it propagates down its side. And remember, what von Hastert said or showed was that one of the modes in which dictyostelium moves is swimming by propagating these little protrusions down its side. Now, this is a 2D version of that, obviously, so it's not the full story, but it's a good step toward, I think, demonstrating that one can begin to understand what is needed to swim in a viscous fluid. Okay, and here's a comparison of how different factors affect the behavior of the two types of swimmers. They're both dictyostelium, but they're slightly different species, and so or varieties, and so they swim slightly differently. Let's see, for example, the efficiency as a function of the maximum protrusion height. In other words, how big a protrusion should you put out of your side in order to swim effectively? Well, for the Barry swimmer, which is a bit more slender than the von Hastert swimmer, the efficiency goes up significantly. Well, they both go up significantly as you increase the height. So it makes sense to put out big protrusions and propagate them down your side. And another comparison is done here, in which you compare a swimmer, which is putting out protrusions at a certain frequency, with a corresponding swimmer who actually alternates them side to side. And what you notice is that this guy actually does have a rotational component to his motion, the same way as you saw in the films of dictyostelium. So the asymmetric protrusions generate the motion, the rotational motion, as well as the linear translation. And the bottom one puts out his protrusions at exactly half the frequency that the top one does, and moves essentially at the same rate as the one who simply alternates. All right, so there are different modes that can be used to achieve the same result. Here, we want to ask, what is the effect of obstacles in the environment? And this is the connection, I think, one of the connections with this workshop, because people used to think that, well, let me back up a minute, because maybe not all of you are familiar with this. One of the key steps in cancer is when the tumor cells begin to metastasize. That is, they begin to move into the surrounding tissue, eventually into the bloodstream, and then circulate throughout the body, and then can invade anywhere. Okay? So an obvious question was, if we can block the metastasis in some way by interfering with how cells move, that should be an effective intervention. So what people did is knock down the integrins, which the cells use to pull themselves through the ECM. Remember the second video or third video that I showed you, which showed you how the cell was pulling on the ECM. So if you knock out those molecules, then you would predict that, well, maybe these cells then don't move. But in fact, what people discovered is that the cells then move, sometimes even more effectively, faster, by simply swimming. Okay? So this is a little attempt at trying to understand what the effect of obstacles on a swimmer might be. So here's a swimmer going past two fixed obstacles. All right? You could think of this as something in the ECM that it has to get past. Compared with a swimmer that's facing no obstacles, obviously there's an effect. Okay? It slows it down somewhat. But if you make the obstacles free, in other words, if the fluid motion can simply push the obstacles aside, then it has very little impact on the motion. You see, this one is almost as efficient as that one. Okay, so that's the exterior problem. Let's look briefly at the interior problem, and that is, what sort of reconstruction of the cytoskeletons are needed to produce the shape changes? Okay, how many people know something about actin dynamics? Okay, let me tell you very quickly for the students. Okay, the cell has within it a kind of network structure which is composed of actin and other polymers. All right. That's what gives it its integrity. Now, you might think that, okay, I have a cell with a certain shape. What do I have to do in order to change that shape significantly, the way it was in the movies? And the answer is, I have to basically reconstruct that internal cytoskeleton, it's called, so as to produce forces where I want to protrude and remove them where I want to remove my motion. So what people have done is try to understand how this actin network can reconstruct itself after some kind of intervention. And it's hard to do in a way that really mimics what happens in a real cell. So what people have done, and this is primarily a group in Germany under Günter Gerisch, is to treat cells with chemicals which knock the actin cytoskeleton down completely. So you imagine this network of polymers, you treat it with something which depolymerizes everything, so the whole internal structure kind of collapses, and then you watch it rebuild after you wash out the treatment. So the question one is trying to answer is, how does a cell generate the forces that it needs to produce those shape changes? Okay? Now, there are two aspects to this, because if you observe cells that are moving in the absence of any signals, you frequently see them moving essentially randomly. So what that tells you is that the dynamical system which is generating those shape changes, in some sense, is generating random motion. So you could think of it perhaps as a chaotic dynamical system. Okay? The shape changes are essentially random because the individual simply pushes protrusions out in various directions to see if they can find suitable directions to move. Okay? If you do this treatment, here's a sequence of stills. What you see is that first the cytoskeleton rebuilds in various points where the cell makes contact with the surface, and then it sometimes generates waves. These waves interact, etc., etc. So the dynamics that result from this treatment are very complex, and here's one example of that. The bright spots that you see are places where the network has been transiently rebuilt. Then you saw a kind of trace of a wave. You'll see another one. There comes another one that starts, builds up, and then propagates through the cell. These waves and these spots only occur where the cell is in contact with the surface. So there's some kind of strong interaction that's not understood with the surface. So if you look at that in stills, here are the stills from that movie. You'll see the various waves that can propagate. And the question is, can one model that effectively, and what does one learn from doing that? Well, that's a very, very complicated process because it involves a large network of biochemical interactions which eventually involve control of actin polymerization and myosin assembly. Okay? So we have made a start on that, and I'll briefly allude to that in a moment, but let me first talk about a slightly simplified version of this. The idea is the following. I told you that these waves and spots only occur where the cell is in contact with the surface, and the consequence of that is that you have some sites on the surface which promote nucleation. Okay? We call those nucleation promoting factors. Okay? Those sites can be activated, and they're actually activated by the network itself, and then they can have a refractory state in which they recover before going back to the unactivated state. And the network itself gives you a positive feedback so that the more filaments you generate, the more you continue to generate. Okay? And here's a kind of diagram of what's involved. So there are these sites on the surface which can be in various conformations. When they're in an active conformation, they can generate a filament. The blue are the actin monomers. The blue, what shall we call them? Rods are the actin filaments. Various molecules are involved. These things are all known, so I won't go through those in detail. But what it leads you to is a very large system of reaction diffusion equations, because you have material diffusing around in the cytoplasm, you have material attaching to the surface, you have material diffusing along the surface. So there's a variety of complicated interactions. And I'm not going to go through these. If you've seen reaction diffusion equations before, then this is fairly standard in that context, except perhaps from the fact that there are fluxes to the boundary and reactions and transport itself on the boundary. But other than that, it's fairly standard. It's a big system of reaction diffusion equations. So what does it... Oh, sorry, here's one more on the nucleation, etc. So yes, there are a number of steps involved. So what does it lead to?
But then you have it's moving, right? Are you looking at when it's moving?
Yeah, I'll show you that it moves.
And what's the condition on the moving boundary?
Hang on. The boundary has a very interesting effect. So another point I didn't make is this is fundamentally a stochastic system. Okay? So we wrote these out as deterministic reaction diffusion equations, but in reality, it's a stochastic process because these sites are individual sites which are then being activated, which nucleate filaments, etc., etc. So we solve it as a stochastic problem, and if I have time, I'll indicate some of the mathematical issues that came up with that. But let's see what that does. whoops, I thought, yes, here's a indication of what happens if you activate a site here in the corner and then watch the front propagate out. Thing that you see immediately is that the system behaves as though it's excitable because there's a wave-like structure, and then behind it the system is in a refractory state. You can see how jagged it is, and it's not only the grid that's making it jagged, it's just the fact that this is fundamentally a stochastic process. If you look at what happens with the colliding waves, it's rather interesting. So you start a wave from each of the corners, and they interact as you would expect, i.e. they annihilate when they reach the center. But it's a stochastic process, and if you remember the movies, you saw in the wake of a wave, you saw occasionally spots reappearing, and then new waves generated from those spots, etc. And you see that here. So we didn't change anything here. We just let the wave move through. But because it is a stochastic process, there are sites which didn't get activated when the first wave went through, but get activated later. And this is exactly what you observe when you see new waves being initiated behind a wave that has passed through. And to answer one aspect of what Othmer was raising, what happens when to this moving boundary problem? Well, the cell is attached to the surface, so there's some fixed boundary that determines the overall domain, and the question is, what happens when the wave gets to the boundary? And the answer here is that it reaches the boundary and it stalls and it dies. The interesting thing experimentally is that you frequently see waves reaching the boundary and then effectively reflecting. They sit there for a while and then they reinitiate at the boundary and propagate toward the interior. We don't have that aspect in there yet. Okay, so how do we link signaling and actin dynamics? Remember, I said there are three aspects to this problem. The exterior problem, the interior problem for how do you generate the shape changes, etc., by changing the cytoskeleton. And what I've shown you here is in no way an answer to that second problem. I mean, don't go away and say that problem's solved. All we've done is do a little bit on the very initial stages of how you would generate the actin network under different conditions, but actually looking at how to translate the actin network into the force needed to move the membrane out is a whole problem in itself, and that hasn't been touched yet. But the problem that links the two is how to get the extracellular signal into the cell. And again, I'm not going to go through this in any kind of detail, but a lot is known about the biochemistry. And I now have a student and I have a postdoc coming who's going to model this process alone in great detail. And the idea is simply that you can think of it as 5 discrete modules of biochemical steps. There's a signal transduction at the very top. There's how that signal gets translated, how it affects the actin dynamics, and so forth. And by looking at it in modular terms, you can understand a great deal about the dynamics of the whole system. I'm not going to go into any detail on that yet, but just show you some preliminary results, again, with a very simplified model. So this is, in essence, the top-level model or module in the signal transduction network, where the signal from the outside gets detected by receptors and it activates something inside. And the something inside is RAS, which is a small g protein, which then goes on to activate other enzymes, etc. And in particular, the enzyme PI3K, which converts 2 different forms of something, and the relative amounts of those are very important. Again, it leads you to a very large kinetic network. And here, incidentally, is a problem that I think you're going to see again and again this week, and that is frequently these biological problems generate large networks, which in itself isn't so bad, but the problem is we know very little about the parameters in those networks. And so a question is, what sort of mathematical techniques can you use to try to identify key steps in those networks so as to give biologists, for example, some guidance as to where they should expend their effort? That would be one thing. Another would be to try and understand whether the networks that are thought to describe phenomena are actually adequate. And I can give you an interesting example in another context of that. So I have a student working on pattern formation in a developmental biology context. And there was a certain network that people thought would explain the observations of what happens when you do a mutation, et cetera, et cetera. What she discovered with a fairly abstract analysis was that that pathway could not predict the observations, and suggested a pathway that would. And then 2 months later, we discovered a paper that just came out last fall which shows that there is exactly this additional network. So it was a beautiful example where she beat the experimentalists, but didn't get it published before they published theirs. But in any case, the mathematical analysis can frequently have a big impact on the understanding of complex networks like this. Okay, anyway, it is complex, but it's not difficult. Oh, well, I shouldn't be quite as pessimistic as I suggested. I said, well, you don't know anything about parameters. You do know some things about parameters. You know bounds, for example, on second-order rate constants. They can't be bigger than something. So it's not completely hopeless. But even within those bounds, if you have 30, 40, or more parameters, you have a huge space to explore. But in this case, it's relatively simple to qualitatively and even quantitatively, in terms of time and relative amplitudes, reproduce some of the observations of the responses of the system to step changes. Okay, let's see. And, well, okay, so one can say something about the differences between the front and back signal. Okay, so in this orientation problem, which is the problem of when a cell gets a signal, how does it decide to go in the correct direction? You have to somehow take a relatively weak external signal and amplify it internally so as to produce the motion in the direction that you want to move, okay? And so the network has to do that, and that's one of the things one has to check to do. And here is what the observations are front to back, and here is what the model predicts front to back for the signals of this particular, in this particular system. Okay, so it can be done. Okay, and an interesting thing which there are some observations on, but is not understood at all, is that the curvature of the membrane locally has a big effect on either suppressing or amplifying the signal. Some people think that there are molecules in the membrane which respond to the local curvature, but what this shows is that that may not be necessary at all. For example, here you see that this signal, whatever it represents, is very high in regions of high curvature and very low along the sides, which is what you want. Okay. See, Philip? 18 after? Okay, so a couple of take-home messages. Surprisingly, or perhaps not surprisingly, cells can display enormous plasticity. That is, they can modify how they move depending on their mechanical interaction with the environment. So there's some feedback from detecting the properties of the environment to the kind of movement scheme that they're going to use. In this 2D version, we were able to look at the types of shape changes that can produce translation. I didn't have time to really go through the efficiency, but one of the things we're really looking at now is the relationship between how fast you propagate those protrusions, their size, the efficiency of the swimming, different measures of the efficiency, for example. What do you think a suitable measure of efficiency might be? Well, if you think in terms of minimum dissipation of energy, that might be an attractive one. How many people would think that that's a good criterion for determining optimal swimming strokes? You want to dissipate minimal energy.
How much energy does a cell uses on movement? Only not very much.
Yeah, but hang on. Pardon?
Also, animals which are subject to predation, they don't want to have a far field. I mean, they don't want to stir the water too much when they are moving on their own.
Or said otherwise, if you're a zebra being chased by a tiger, you're not worried about efficiency. You just want to get the hell out of there, right? So there are different criteria depending on what you're trying to accomplish. So, for example, if you were to say, let's use minimum energy dissipation, then the answer is don't move. You don't dissipate any energy, right? So you really have to think. And it's not obvious what criteria should be used. And so analysis of different possibilities may be useful. Okay, so this is work with all the swimming with Qishuan Wang and the networks, etc., the actin reconstruction with Ranyu Kambavath and Jifeng Hu. Ranyu saw the light. He's now working at Goldman Sachs in London. So thank you.